Long before humans ever dreamed of capturing sunlight to power their world, nature had already perfected the art. Every leaf, every blade of grass, every microscopic algae floating in the ocean is a living solar panel, quietly and elegantly converting light into life itself. This ancient technology, refined over billions of years, doesn't just sustain plants, it sustains everything. Every breath you take, every meal you eat, every moment of your existence traces back to this gentle green miracle happening all around us, so softly and continuously that we barely notice it at all. Hi, I'm Luna, and if you find comfort in quiet curiosity, gentle science, and stories that drift like starlight, you're in just the right place. So wrap yourself in something warm, settle in. And if you'd like to hear more sleepy wonderings, feel free to like and subscribe because tonight, we're going to explore the most peaceful revolution that ever happened on Earth. A revolution that turned our planet green, filled our skies with oxygen, and made complex life possible. We'll drift through the microscopic worlds where light becomes sugar, where water splits into its component parts with the gentleness of morning dew, and where ancient molecular machinery works with a precision that would make our finest engineers weep with admiration. As you settle deeper into comfort, imagine for a moment that you could shrink down small enough to witness this miracle firsthand. Picture yourself floating gently through the green corridors of a leaf, watching as particles of sunlight arrive like tiny golden messengers, each one carrying just enough energy to power the most elegant chemistry our world has ever known. This is photosynthesis, quiet, constant, beautiful, and tonight, we're going to understand how it works, why it matters, and what it teaches us about the patient, persistent power of life itself. To understand photosynthesis, we need to begin where all stories of light begin, with the sun itself. Every morning, our star sends out a gentle flood of electromagnetic radiation, packets of energy we call photons, streaming across the vastness of space at the speed of light. These photons began their journey in the sun's core, where temperatures reach 15 million degrees Celsius, and hydrogen atoms fuse together to create helium in a dance of nuclear fire. But by the time these photons reach us, having traveled 93 million miles through the cold vacuum of space, they arrive as gentle warmth on our skin, as the golden light that makes everything visible, as the invisible energy that powers every living thing on our planet. Plants have learned to catch these traveling photons like fishermen casting nets into a river of light. But unlike our clumsy solar panels, which convert only about 20% of sunlight into usable electricity, plants have developed something far more elegant. They don't just capture light, they weave it into the very fabric of life, transforming it into sugars that can be stored, transported, and used to build everything from the tiniest seedling to the mightiest redwood tree. The process begins in structures so small they exist at the very edge of what we can imagine, chloroplasts. Tiny green organelles floating within plant cells, like miniature factories, dedicated to the art of light harvesting. Each chloroplast contains hundreds of thousands of chlorophyll molecules, those remarkable green pigments that give plants their color and their power. Chlorophyll is a molecule of such elegant complexity that it seems almost designed by nature's finest architect. A ring of carbon and nitrogen atoms, cradling a single atom of magnesium at its heart, with long hydrocarbon tails that anchor it to the protein structures around it. But chlorophyll doesn't work alone. It's surrounded by a constellation of helper molecules, carotenoids that capture light at different wavelengths, proteins that channel energy with perfect efficiency, and enzymes that orchestrate chemical reactions with a precision that makes Swiss clockwork seem crude by comparison. Together, these molecules form photosystems, molecular machines that have been operating continuously for over 3 billion years, quietly converting light into life with an efficiency that our best technology can barely match. When a photon strikes a chlorophyll molecule, something beautiful happens. The energy of light excites an electron, lifting it to a higher energy state like a note rising to a higher pitch on a musical scale. But this isn't chaos, it's choreography. The excited electron doesn't just bounce around randomly. Instead, it's passed along a chain of molecules like a gentle relay race, each handoff perfectly timed and precisely controlled. This is the beginning of photosynthesis, the moment when the energy of starlight becomes the energy of life. As you rest more deeply, imagine the billions upon billions of these light capture events happening in the leaves outside your window right now. Even in the gentle moonlight, some plants continue their quiet work, processing the day's stored energy, growing imperceptibly in the darkness. This is the patient persistence of photosynthesis, not dramatic or sudden, but constant and reliable as the sunrise itself. The chemistry of photosynthesis can be written in a simple equation that belies its stunning complexity. 6 molecules of carbon dioxide plus 6 molecules of water, combined with the energy of sunlight, yield 1 molecule of glucose sugar and 6 molecules of oxygen. Written out, it looks almost elementary. 6CO2 plus 6H2O plus light energy right arrow C6H 12O6 plus 6O2. But within this simple formula lies one of the most sophisticated chemical processes ever discovered, a process that required nearly 2 billion years of evolution to perfect. Consider for a moment the elegance of this transformation. Carbon dioxide, a gas that we exhale as waste, becomes the carbon backbone of sugar molecules. Water, the simplest and most abundant molecule on our planet, provides the hydrogen that will help form organic compounds and releases oxygen as a byproduct. And sunlight, pure energy racing across space from our nearest star, provides the power to make it all happen. It's as if nature found a way to weave together the most basic elements of our world, air, water, and light, into the complex chemistry of life itself. But photosynthesis is actually 2 processes working in perfect harmony, like 2 movements of a gentle symphony. The first movement, called the light-dependent reactions, or the photo part of photosynthesis, captures light energy and uses it to split water molecules. This happens in the thylakoid membranes within chloroplasts, where chlorophyll and its helper molecules are arranged in precise patterns like solar cells in a panel. When light energy excites electrons in chlorophyll, those electrons are passed along an electron transport chain, releasing energy at each step. This energy is used to pump hydrogen ions across the thylakoid membrane, creating a concentration gradient that drives the production of ATP, the universal energy currency of life. Meanwhile, the splitting of water molecules, a process called photolysis, releases hydrogen ions, electrons to replace those lost by chlorophyll, and oxygen as a byproduct. This oxygen diffuses out of the leaf and into the atmosphere, where it joins the great reservoir of breathable air that surrounds our planet. Every second breath you take was produced by photosynthesis, released by some plant somewhere as it quietly went about the business of converting light into life. The second movement of photosynthesis, called the light-independent reactions, or the Calvin cycle, takes place in the stroma of the chloroplast. Here, carbon dioxide from the atmosphere is fixed into organic molecules through a process discovered by Melvin Calvin in the 1950s. Using the ATP energy generated during the light reactions, carbon dioxide molecules are incorporated into larger organic compounds through a series of reactions so elegantly orchestrated that each step leads inevitably to the next. like phrases in a perfectly composed melody. The Calvin cycle begins with an enzyme called Rhu-bis-Co, probably the most abundant protein on Earth, and certainly one of the most important. Rhu-bis-Co catalyzes the reaction between carbon dioxide and a 5-carbon sugar called Rhu-BP, creating an unstable 6-carbon compound that immediately splits into 2 3-carbon molecules. These molecules then undergo a series of transformations, powered by the ATP and NADPH generated during the light reactions, eventually producing glucose and regenerating the Rhu-BP needed to keep the cycle running. It's a process of remarkable efficiency and elegance, fine-tuned by billions of years of evolution to extract carbon from the atmosphere and incorporate it into the living world. Each turn of the Calvin cycle fixes one molecule of CO2, and it takes 6 turns to produce 1 molecule of glucose. The process continues as long as light is available to power the light reactions, and as long as carbon dioxide is available to fuel the carbon-fixing reactions. As these two processes work together, light capture and carbon fixation, they create something profound. The transformation of inorganic matter into organic matter, the conversion of simple molecules into complex sugars, the weaving of sunlight into the fabric of life itself. This is the fundamental miracle that makes everything else possible. The quiet revolution that turned our planet from a barren rock into a living world. To truly appreciate photosynthesis, we need to journey back in time, far back to when our planet was young and the chemistry of life was just beginning to unfold. Picture Earth as it was nearly 4 billion years ago, a world without flowers, without forests, without the gentle green that now covers our continents. The atmosphere contained almost no oxygen, the sky was a different color, and the only life existed in the form of simple microbes floating in ancient seas. These early microbes had learned to harness chemical energy from their environment, breaking down sulfur compounds, metabolizing methane, extracting energy from iron. But they were limited by the available chemicals around them, like miners working a finite vein of ore. Then, sometime between 3.5 and 2.5 billion years ago, certain bacteria made a discovery that would change everything. They learned to use the energy of sunlight to power their cellular processes, not by complex photosynthesis as we know it today, but through simpler forms of light harvesting. These early photosynthetic bacteria were pioneers in the truest sense, exploring new ways to capture and use energy. Some used hydrogen sulfide instead of water, producing sulfur compounds instead of oxygen. Others learned to harness different wavelengths of light, developing purple and green pigments that could capture photons invisible to our eyes. They were experimenting with the fundamental chemistry of light and life, laying the groundwork for the more sophisticated systems that would follow. But the real revolution began with a group of bacteria called cyanobacteria, sometimes known as blue-green algae, though they're not actually algae at all. These remarkable microbes were the first to perfect what we call oxygenic photosynthesis, the process of using water as an electron donor and producing oxygen as a byproduct. It seems like such a simple innovation, but it required solving an enormous chemical challenge. Water is a very stable molecule, and splitting it requires a tremendous amount of energy. The cyanobacteria had to evolve molecular machinery powerful enough to tear apart water molecules while still capturing and storing the energy released. The solution they developed was elegant in its complexity. They created Photosystem II, a molecular machine that could harness light energy to generate the electrical potential needed to split water. This photosystem contains a cluster of 4 manganese atoms and 1 calcium atom, arranged in a precise geometric pattern that acts like a microscopic battery. When energized by light, this manganese-calcium cluster accumulates enough electrical charge to rip electrons away from water molecules, breaking the chemical bonds that hold H2O together. This breakthrough happened gradually, over hundreds of millions of years. But once cyanobacteria mastered oxygenic photosynthesis, they began to spread across the ancient oceans like a green tide. They could live anywhere sunlight reached water, from shallow pools to the open sea. They formed great mats and stromatolites, layered structures that we can still see fossilized in rocks today, silent testimony to the ancient revolution that was quietly transforming our planet. As these cyanobacteria multiplied and spread, they began pumping oxygen into an atmosphere that had never known it before. This wasn't a gentle process by geological standards. It was rapid and dramatic, like adding a new chemical ingredient to a complex recipe. The oxygen they produced was initially absorbed by dissolved iron in the oceans, creating bands of rust-colored iron oxide that we can still see in rock formations around the world. These banded iron formations are like pages in Earth's diary, recording the moment when photosynthesis began to change the chemistry of our planet. But eventually, the iron was used up, and oxygen began to accumulate in the atmosphere itself. This created what scientists call the Great Oxidation Event, also known as the oxygen catastrophe. Catastrophe because oxygen was actually toxic to most life forms that existed at the time. Many ancient microbes that had thrived in the oxygen-free world died out, unable to cope with this new reactive gas that could damage their cellular machinery. Yet this apparent catastrophe was actually the foundation for all future complexity. Oxygen is incredibly reactive, which makes it dangerous, but also incredibly useful. Organisms that learn to harness oxygen could extract far more energy from their food than those limited to oxygen-free metabolism. Aerobic respiration, the process of using oxygen to break down organic molecules, yields about 18 times more energy than anaerobic processes. This energy windfall made possible the evolution of larger, more complex cells, and eventually multicellular organisms. The accumulation of oxygen in the atmosphere also led to the formation of the ozone layer. As some of that oxygen was transformed by ultraviolet light into ozone molecules, this created a protective shield high in the atmosphere that blocked harmful UV radiation from reaching Earth's surface, making it safer for life to venture out of the protective depths of the oceans and eventually colonize the land. As you settle deeper into rest, imagine the patience of this transformation. It took hundreds of millions of years for cyanobacteria to change the chemistry of our planet's atmosphere. Generation after generation of these microscopic organisms lived and died, each one contributing its tiny portion of oxygen to the growing reservoir in the sky. There was no plan, no consciousness driving this change, just the simple act of photosynthesis, repeated billions of times across millions of centuries, quietly and persistently transforming a world. The descendants of those ancient cyanobacteria are still with us today. Some still live as free-floating bacteria in our oceans and lakes, continuing the work their ancestors began billions of years ago. But others took a different path, one that led to an even more remarkable transformation. Around 1.5 billion years ago, some early eukaryotic cells with nuclei, like the ones that make up plants and animals, engulfed cyanobacteria in a process that wasn't quite eating and wasn't quite infection. Instead of digesting these bacterial prisoners, the larger cells formed partnerships with them. This was the beginning of endosymbiosis, one of the most important evolutionary innovations in Earth's history. the cyanobacteria became chloroplasts, permanent residents within plant cells, retaining their ability to photosynthesize while receiving protection and nutrients from their hosts. In return, they shared the sugars they produced from sunlight with their host cells. It was a partnership that benefited both sides so much that it became permanent, locked in by millions of years of coevolution. This endosymbiotic relationship explains why chloroplasts have their own DNA, separate from the plant cell's nuclear DNA. They still reproduce independently, dividing like bacteria within their plant hosts. When you look at the genetic code of chloroplasts, you can see their bacterial ancestry written clearly in their DNA sequences. They are living fossils, ancient microbes that found a new way to exist within the cells of plants. The partnership between plant cells and their chloroplast residents proved so successful that it gave rise to all the photosynthetic complexity we see today. Green algae floating in ponds, vast kelp forests swaying in ocean currents, towering trees reaching toward the sky, delicate flowers opening to morning sunlight. All of them are powered by the descendants of ancient cyanobacteria that learned to split water molecules and release oxygen into the world. Within each leaf, within each blade of grass, within every photosynthetic cell, there exists an architecture of stunning sophistication designed entirely around the capture and processing of light. To understand how intricate this system is, imagine if we tried to build a factory that could operate using only sunlight as its power source, converting carbon dioxide from the air into useful products, and doing so with near-perfect efficiency, while also producing oxygen as a beneficial byproduct. The engineering challenges would be staggering. Yet plants accomplish this feat in structures so small that hundreds could fit within the period at the end of this sentence. The chloroplast itself is a marvel of biological architecture, roughly lens-shaped and typically measuring just a few micrometers across. Yet within this tiny space, nature has packed an incredibly sophisticated light harvesting and energy conversion system. The chloroplast is enclosed by two membranes, creating a protected internal environment where the delicate chemistry of photosynthesis can proceed without interference. Inside this envelope, the chloroplast is filled with a gel-like substance called the stroma, where the Calvin cycle takes place, and where many of the chloroplast's own genes are stored and transcribed. But the most remarkable structures within the chloroplast are the thylakoids, flattened, disc-like sacs that are stacked on top of each other like coins in a roll. These stacks are called grana, and they give chloroplasts their distinctive internal structure when viewed under a microscope. The thylakoid membranes are where the light harvesting apparatus is housed, arranged in precise patterns that maximize the capture and utilization of solar energy. Embedded within these thylakoid membranes are the photosystems, molecular complexes that contain chlorophyll and other pigments, along with the proteins and enzymes needed to convert light energy into chemical energy. There are two main types of photosystems, creatively named photosystem I and photosystem II. Though somewhat confusingly, photosystem II actually operates first in the sequence of reactions. Each photosystem is a masterpiece of molecular engineering, containing hundreds of individual molecules working together with precision that rivals our finest machines. Photosystem II is perhaps the most remarkable molecular machine on Earth. It contains the oxygen-evolving complex, the 4-manganese-1-calcium cluster that performs the seemingly impossible task of splitting water molecules. This cluster is surrounded by proteins that hold it in precisely the right position and protect it from damage, while channels in the protein structure allow water molecules to approach the cluster and oxygen molecules to escape. The entire system operates like a microscopic water-splitting engine, powered by sunlight and engineered by evolution to operate continuously for months or years without failure. The chlorophyll molecules within the photosystems aren't randomly scattered, but are arranged in precisely organized antenna complexes that function like natural satellite dishes designed to capture light. These antenna complexes contain not just chlorophyll, but also carotenoids, yellow, orange, and red pigments that can absorb wavelengths of light that chlorophyll misses. Together, these pigments create a light harvesting network that can capture photons across a broad spectrum of visible light and efficiently funnel that energy toward the reaction centers where photochemistry takes place. The efficiency of this light harvesting system is breathtaking. When a photon is absorbed by any pigment molecule in an antenna complex, the energy is passed from molecule to molecule through a process called resonance energy transfer until it reaches the reaction center chlorophyll. This transfer happens with nearly 100% efficiency and takes only a few picoseconds, trillionths of a second. It's as if the antenna complex were a perfectly designed funnel for light energy, gathering photons from a relatively large area and concentrating them precisely where they're needed. Once energy reaches the reaction center, the real photochemistry begins. In photosystem II, the reaction center chlorophyll molecule absorbs the concentrated light energy and uses it to eject an electron, which is immediately captured by a nearby electron acceptor molecule. This creates what chemists call a charge separation. The reaction center chlorophyll becomes positively charged, while the electron acceptor becomes negatively charged. It's like creating a microscopic battery, with positive and negative poles separated by just a few nanometers. But this charge separation would be useless if it immediately reversed, with the electron falling back to the positively charged chlorophyll. To prevent this, evolution has created an elegant solution: a series of electron acceptor molecules that form a one-way chain, passing the electron along from one to the next and preventing it from returning to its origin. Each step in this electron transport chain releases a small amount of energy, which is captured and used to pump hydrogen ions across the thylakoid membrane. This proton pumping creates an electrochemical gradient across the thylakoid membrane, with a high concentration of hydrogen ions inside the thylakoid space and a lower concentration in the stroma. Think of it as creating a microscopic dam, with hydrogen ions piling up on one side of the membrane like water behind a dam wall. This gradient represents stored energy, potential energy, that can be released when the hydrogen ions are allowed to flow back across the membrane. The mechanism that harnesses this gradient is one of the most elegant molecular machines in biology: ATP synthase. This protein complex spans the thylakoid membrane and acts like a tiny turbine, using the flow of hydrogen ions to drive the synthesis of ATP from ADP and phosphate. As hydrogen ions flow through ATP synthase from the high concentration side to the low concentration side, they cause parts of the protein to rotate like the shaft of a turbine. And this rotation drives the chemical reactions that produce ATP. Meanwhile, the electrons that have traveled through the electron transport chain are passed to photosystem I, where they absorb another dose of light energy and are boosted to an even higher energy level. These high-energy electrons are then used to reduce NADP+ to NADPH, creating another form of chemical energy that will be used in the Calvin cycle. The entire system operates like a sophisticated chemical factory powered by sunlight, with raw materials entering at one end and useful products emerging at the other. But what about the positively charged reaction center, chlorophyll, in photosystem II. It needs electrons to replace the ones it has lost. And this is where the water-splitting complex comes into play. The manganese-calcium cluster uses light energy to extract electrons from water molecules, breaking apart H2O to produce hydrogen ions, electrons, and oxygen. The electrons replace those lost by the reaction center chlorophyll. The hydrogen ions contribute to the proton gradient that drives ATP synthesis, and the oxygen diffuses out of the chloroplast and eventually out of the plant as a waste product that happens to be essential for life on Earth. As you rest in the gentle darkness, consider the symphony of molecular motions happening in the plants around you. Even now, in dim light or stored energy, the molecular machinery of photosynthesis continues its work. Electrons are flowing through transport chains with the regularity of rivers flowing to the sea. ATP synthase turbines are spinning as hydrogen ions flow through them like water through mill wheels. The ancient partnership between plant cells and their bacterial residents continues the patient work of converting inorganic matter into living tissue. The precision required for all of this to work is extraordinary. The distances between electron transport molecules must be exact, too far apart, and electrons can't jump from one to the next. Too close, and the system becomes unstable. The orientation of pigment molecules must be precise. Each chlorophyll molecule positioned exactly right to pass energy efficiently to its neighbors. The protein structures must be flexible enough to accommodate the movement of molecules, yet rigid enough to maintain their function over months of continuous operation. This level of precision extends to the temporal coordination of the system as well. The light reactions of photosynthesis operate on timescales from picoseconds to milliseconds, with each step precisely timed to occur when the previous step has been completed. The Calvin cycle operates on timescales from milliseconds to seconds, with its reactions synchronized to the availability of ATP and NADPH from the light reactions. And the overall regulation of photosynthesis responds to changes in light intensity, temperature, and carbon dioxide levels, with response times from seconds to minutes. Plants have also evolved sophisticated mechanisms to protect their photosynthetic apparatus from damage. Too much light can actually harm the delicate molecular machinery of photosynthesis. So plants have developed systems to dissipate excess energy safely. Carotenoid molecules can absorb excess energy and release it as harmless heat. Entire photosynthetic complexes can be taken offline temporarily when light levels are too high. Some plants can even adjust the angle of their leaves to avoid excess light exposure during the hottest part of the day. The architecture of photosynthesis extends beyond individual chloroplasts to the structure of leaves themselves. The arrangement of cells within a leaf is optimized for light capture and gas exchange. The upper epidermis is transparent, allowing light to penetrate to the photosynthetic cells below. The palisade mesophyll cells just under the upper surface are packed with chloroplasts and arranged like columns to maximize light absorption. The spongy mesophyll below contains air spaces that facilitate the diffusion of carbon dioxide to photosynthetic cells and the removal of oxygen. Even the shape and orientation of leaves reflect the demands of efficient photosynthesis. Broad, flat leaves maximize the surface area available for light capture. Leaf orientation can be adjusted throughout the day to track the sun's movement across the sky. The waxy cuticle that covers leaf surfaces allows light to pass through while preventing water loss. Stomata, tiny pores in the leaf surface, can open and close to regulate the exchange of carbon dioxide and oxygen while controlling water loss. If we could somehow view our planet from space with eyes sensitive to the flow of energy and matter, we would see Earth as a vast, interconnected system of photosynthetic activity, from the microscopic phytoplankton floating in our oceans to the towering canopies of our forests. Photosynthesis operates as a global engine that drives the chemistry of life on a planetary scale. Every day, plants and photosynthetic microbes capture approximately 100 terawatts of solar energy, roughly 6 times more energy than all of human civilization uses in a year, and transform it into organic compounds that fuel the entire web of life. This global photosynthetic engine processes enormous quantities of matter as well as energy. Each year, photosynthesis removes approximately 120 billion tons of carbon dioxide from our atmosphere and incorporates it into organic molecules. To put this in perspective, this is equivalent to processing the entire mass of carbon dioxide in the atmosphere every 7 years. At the same time, photosynthesis releases about 100 billion tons of oxygen annually, maintaining the oxygen-rich atmosphere that makes complex life possible. The scale of this global operation becomes even more impressive when we consider that it's performed by organisms ranging from single-celled diatoms to massive sequoia trees, each one operating the same basic biochemical machinery, but adapted to their particular environments. In the open ocean, microscopic phytoplankton, floating plant-like organisms, carry out photosynthesis near the surface where sunlight can penetrate. These tiny organisms are so numerous and so active that they produce more than half of the oxygen we breathe, despite being invisible to the naked eye. Ocean photosynthesis has its own unique characteristics and challenges. Seawater absorbs different wavelengths of light at different rates, with red light disappearing within the first few meters, while blue light can penetrate to depths of several hundred meters. Marine photosynthetic organisms have adapted to these conditions by evolving pigments that can efficiently capture the blue-green light available in their underwater world. Some have developed specialized pigment combinations that allow them to photosynthesize effectively, even in the dim light of deeper waters. The seasonal cycles of ocean photosynthesis are driven largely by nutrient availability rather than light levels. In many parts of the ocean, the nutrients needed for photosynthesis, particularly nitrogen and phosphorus, are most abundant during certain times of the year when deep, nutrient-rich waters are brought to the surface by currents or seasonal mixing. This creates dramatic blooms of phytoplankton that can be seen from space, turning vast areas of ocean green with photosynthetic activity. These phytoplankton blooms are the foundation of ocean food webs, converting sunlight and dissolved nutrients into organic compounds that feed everything from tiny copepods to massive whales. When phytoplankton die, some of their organic matter sinks to the ocean depths, carrying carbon away from the atmosphere and storing it in deep ocean sediments. This biological carbon pump is one of the most important mechanisms for regulating Earth's climate, removing carbon dioxide from the atmosphere and storing it in the deep ocean for hundreds to thousands of years. On land, photosynthesis takes on different forms adapted to the tremendous variety of terrestrial environments. In tropical rainforests, where sunlight and water are abundant year-round, plants engage in a fierce competition for light, leading to the evolution of complex forest canopies with multiple layers of photosynthetic activity. The upper canopy trees capture most of the direct sunlight, while understory plants have evolved to photosynthesize efficiently in the filtered, dappled light that reaches the forest floor. These forest ecosystems represent some of the most intensive photosynthetic activity on the planet. A single hectare of tropical forest can contain hundreds of tree species, thousands of other plant species, and countless photosynthetic microorganisms, all working together to capture and process solar energy. The total leaf surface area in a forest, called the leaf area index, can be several times larger than the ground area, as multiple layers of leaves capture light at different levels of the canopy. In temperate regions, photosynthesis follows seasonal rhythms driven by changes in temperature and daylight hours. Deciduous trees have evolved the strategy of growing new leaves each spring, maximizing their photosynthetic capacity during the warm, bright months of summer, then dropping their leaves in autumn to conserve resources during the cold, dark months of winter. This seasonal cycle is so synchronized with local climate conditions that the timing of leaf emergence and senescence can be used as an indicator of climate change. Grasslands represent a different strategy for terrestrial photosynthesis. Rather than competing for height to capture light, grasses focus their photosynthetic activity close to the ground and invest heavily in extensive root systems that can quickly capture nutrients and water. Prairie grasses can have root systems that extend several meters deep and contain more biomass underground than above ground. This strategy makes grasslands incredibly efficient at capturing and storing carbon, with prairie soils containing some of the richest carbon deposits on Earth. In arid environments, plants have evolved remarkable adaptations to maintain photosynthesis despite limited water availability. Succulent plants like cacti use a specialized form of photosynthesis called CAM photosynthesis, where carbon dioxide is captured and stored at night when temperatures are cool and humidity is higher, then used for photosynthesis during the day when the plant's stomata can remain closed to prevent water loss. This temporal separation of gas exchange and light capture allows these plants to photosynthesize in environments where water is so scarce that other plants cannot survive. Some plants in hot, arid climates use a different strategy called C4 photosynthesis, which concentrates carbon dioxide around the Calvin cycle enzymes to improve the efficiency of carbon fixation under hot, dry conditions. C4 plants include many of our most important crop species: corn, sugarcane, sorghum, and some of our most productive grassland species. These plants can maintain high rates of photosynthesis even when water stress forces them to partially close their stomata, making them well adapted to hot, dry climates. Arctic and alpine plants face the challenge of photosynthesizing under extreme cold and during very short growing seasons. These plants have evolved strategies to begin photosynthesis almost immediately when conditions become favorable, often growing and photosynthesizing under snow or in temperatures that would kill plants from warmer climates. Some Arctic plants have developed the ability to maintain their photosynthetic machinery in a state of readiness throughout the winter, allowing them to begin photosynthesis as soon as sunlight becomes available in spring. The global engine of photosynthesis also includes countless epiphytes, plants that grow on other plants, from Spanish moss-draping southern trees to colorful bromeliads adorning tropical tree trunks. These plants have evolved to capture nutrients and moisture from the air itself, carrying out photosynthesis while perched high in the canopy where light is abundant, but soil is non-existent. As you settle deeper into peaceful rest, consider the vast, interconnected network of photosynthetic activity surrounding our planet at this very moment. Somewhere in the world, it's always daylight, and wherever the sun shines on green plants or photosynthetic microbes, the ancient machinery of light capture continues its patient work. Ocean currents carry phytoplankton into areas of bright sunlight where they burst into photosynthetic activity. Desert plants are using the cool of night to capture carbon dioxide for tomorrow's photosynthesis. Forest trees are adjusting their leaf angles to track the sun's movement across the sky. This global photosynthetic engine operates with a resilience and redundancy that human technology has yet to match. If one ecosystem is damaged by fire, flood, or drought, other ecosystems continue their photosynthetic work, maintaining the global cycles of carbon and oxygen that keep our planet habitable. The system is so robust and so distributed that it has maintained Earth's atmospheric composition within the narrow range, suitable for complex life for hundreds of millions of years. Despite volcanic eruptions, asteroid impacts, ice ages, and other massive disruptions, the interconnectedness of global photosynthesis extends to the movement of nutrients and organic compounds around the planet. Carbon fixed by photosynthesis in one location can be transported vast distances by rivers, ocean currents, and atmospheric circulation before being returned to the carbon cycle. Organic compounds produced by phytoplankton in the ocean can wash up on distant shores, fertilizing terrestrial ecosystems. Dead plant matter from forests can be carried by rivers to the deep ocean, where it provides food for deep-sea communities that never see sunlight. Even the seasonal changes in global photosynthesis can be detected from space, as atmospheric carbon dioxide levels fluctuate with the growth and dormancy of northern hemisphere vegetation. During the northern summer, when forests and grasslands are most active, global CO2 levels drop slightly as photosynthesis removes carbon from the atmosphere. During winter, when photosynthesis slows and decomposition continues, CO2 levels rise slightly. This seasonal breathing of the planet is testimony to the enormous scale of photosynthetic activity and its impact on global atmospheric chemistry. Deep within the quiet interior of every photosynthesizing cell, a molecular dance of exquisite precision unfolds continuously, so complex and coordinated that it rivals the most sophisticated ballet ever choreographed. This is the Calvin cycle, the metabolic pathway through which plants capture carbon dioxide from the atmosphere and weave it into the organic molecules that form the foundation of all life. To witness this process at the molecular level would be to see chemistry as poetry, each reaction flowing inevitably into the next, guided by enzymes that have been perfected over billions of years of evolution. The Calvin cycle takes place in the stroma of chloroplasts, the gel-like matrix that surrounds the thylakoid membranes where light energy is captured. Here, dissolved in the aqueous environment of the stroma, float the enzymes and metabolites that will transform inorganic carbon into organic compounds. The cycle requires the ATP and NADPH produced by the light reactions of photosynthesis, but it doesn't directly require light itself. This is why it's sometimes called the light-independent reactions, though it depends entirely on the products of light-dependent processes. The cycle begins with carbon fixation, the moment when gaseous carbon dioxide from the atmosphere becomes incorporated into an organic molecule. This crucial step is catalyzed by an enzyme called Ru-bis-Co, ribulose 15-bisphosphate carboxylase/oxygenase, which may well be the most important protein on Earth. Ru-bis-Co is certainly the most abundant, making up nearly half the protein content of most plant leaves. And it's estimated that there are about 40 pounds of Ru-bis-Co for every person on the planet. Ru-bis-Co catalyzes the reaction between carbon dioxide and a 5-carbon sugar called ribulose 15-bisphosphate, or Ru-BP. When these two molecules meet at the active site of Ru-bis-Co, something remarkable happens. The stable, gaseous CO2 molecule is captured and combined with the organic Ru-BP molecule, to form an unstable 6-carbon intermediate. This intermediate exists for only the briefest moment before it splits apart into two 3-carbon molecules called 3-phosphoglycerate, or PGA. This might seem like a simple reaction, but it represents one of the most significant chemical transformations on our planet. In this moment, inorganic carbon becomes organic carbon. A molecule that was floating freely in the atmosphere becomes locked into the biochemical machinery of life. Every carbon atom in your body, in every leaf, in every living thing on Earth, passed through this reaction at some point in its journey from the atmosphere to the living world. The 3-phosphoglycerate molecules produced by Rue Biscoe are the raw materials for everything that follows. Through a series of carefully orchestrated reactions, these simple 3-carbon compounds will be transformed into the complex organic molecules that plants need to grow and survive. But first, they must be activated using the energy captured from sunlight. The activation process, called the reduction phase of the Calvin cycle, uses both ATP and NADPH produced by the light reactions. Each molecule of 3-phosphoglycerate is first phosphorylated by ATP, adding a phosphate group that makes the molecule more reactive. The resulting compound, 13-bisphosphoglycerate, is then reduced by NADPH, which donates electrons and hydrogen to convert it into glyceraldehyde 3-phosphate, or G3P. G3P is a crucial molecule in plant metabolism. It's the primary product of photosynthesis and the starting point for the synthesis of glucose, starch, cellulose, and countless other organic compounds. Two molecules of G3P can be combined to form one molecule of glucose, providing the sugar that plants use for energy and growth. Some G3P is immediately used for cellular metabolism, while some is converted into starch for energy storage, and some is used to synthesize the cellulose and other structural materials that make up plant tissues. But here's where the elegant choreography of the Calvin cycle becomes most apparent. For the cycle to continue, some of the G3P molecules must be used to regenerate the Rue BP molecules that serve as acceptors for new CO2 molecules. This regeneration phase is perhaps the most complex part of the Calvin cycle, involving a series of reactions that rearrange and recombine G3P molecules to produce Rue BP. The mathematics of the Calvin cycle are beautiful in their precision. For every 3 molecules of CO2 that enter the cycle, 6 molecules of 3PGA are produced, which are then converted into 6 molecules of G3P. One of these G3P molecules represents the net gain of carbon. It can be used to synthesize glucose or other compounds. The remaining 5 G3P molecules are rearranged through a series of reactions to regenerate 3 molecules of Rue BP, allowing the cycle to continue. This regeneration process involves a complex series of enzymatic reactions that rearrange carbon skeletons like a molecular puzzle. 3-carbon compounds are combined to form 4-carbon compounds, which are then rearranged to form 5-carbon compounds, which are converted back into Rue BP. Each reaction is catalyzed by a specific enzyme that recognizes particular molecular structures and catalyzes particular bond-breaking and bond-forming reactions. The entire process requires a tremendous investment of energy. To fix 3 molecules of CO2 and produce 1 molecule of G3P requires 9 molecules of ATP and 6 molecules of NADPH. This represents a substantial portion of the energy captured by the light reactions of photosynthesis, highlighting just how energy expensive it is to convert inorganic carbon into organic compounds. Despite this energy cost, or perhaps because of it, plants have evolved sophisticated mechanisms to regulate the Calvin cycle and ensure it operates efficiently. The key enzymes of the cycle are activated by light, either directly through reduction by electrons from the photosynthetic electron transport chain, or indirectly through changes in pH and magnesium concentrations that occur when the light reactions are active. This ensures that the energy-expensive Calvin cycle operates only when the light reactions are producing the ATP and NADPH needed to power it. Plants also regulate the Calvin cycle in response to the availability of CO2. When CO2 concentrations are high, Rubis CO operates efficiently, fixing carbon and producing G3P. But when CO2 concentrations are low, which often happens when plants close their stomata to conserve water, Rubis CO can react with oxygen instead of CO2, leading to a wasteful process called photorespiration. Photorespiration is essentially the opposite of photosynthesis. Instead of fixing CO2 and producing organic compounds, it consumes organic compounds and releases CO2. It occurs because Rubis CO evolved billions of years ago when atmospheric oxygen levels were much lower than they are today. The enzyme can't distinguish perfectly between CO2 and O2. And when the ratio of CO2 to O2 is low, it sometimes catalyzes the reaction of RuBP with oxygen instead of carbon dioxide. This evolutionary legacy creates problems for plants, especially in hot, dry conditions when stomata must be kept closed to prevent water loss. Under these conditions, CO2 levels inside leaves drop while O2 levels rise, favoring photorespiration over photosynthesis. Some plants have evolved mechanisms to minimize photorespiration, including the C4 and CAM, photosynthetic pathways that concentrate CO2 around Rubis CO. C4 photosynthesis involves an additional carbon fixation step that occurs before the Calvin cycle. CO2 is first fixed into a 4-carbon compound by an enzyme called PEP carboxylase, which doesn't react with oxygen. This 4-carbon compound is then transported to specialized cells where CO2 is released and concentrated around Rubis CO, creating conditions that favor photosynthesis over photorespiration. This spatial separation of initial CO2 fixation and the Calvin cycle allows C4 plants to maintain high rates of photosynthesis even when CO2 levels are low. CAM photosynthesis achieves the same result through temporal rather than spatial separation. CO2 is captured and stored in organic acids at night, when stomata can be open without excessive water loss. During the day, these organic acids are broken down to release CO2 internally, allowing photosynthesis to proceed with stomata closed. This strategy is particularly common in desert plants, where water conservation is critical for survival. As you rest in the gentle darkness, imagine the countless Calvin cycles operating in the plants around you. Even now, in darkness or dim light, some of the enzymes involved in carbon fixation remain active, processing the last remnants of CO2 captured during daylight hours. The molecular machinery continues its patient work, transforming atmospheric carbon into the organic compounds that will become new leaves, new roots, new growth when spring returns. The precision required for the Calvin cycle to operate efficiently is extraordinary. Each enzyme must recognize its specific substrate molecules among thousands of other compounds present in the cell. The binding sites on these enzymes are shaped to fit their substrate molecules like locks fit keys, ensuring that each reaction occurs at the right time and in the right place. The regulation of enzyme activity must be perfectly coordinated with the light reactions and with the plant's overall metabolic needs. This coordination extends to the subcellular level as well. The Calvin cycle enzymes are not randomly distributed throughout the stroma. but are organized into complexes and microcompartments that facilitate the efficient flow of metabolites from one reaction to the next. Some enzymes are associated with the surfaces of thylakoid membranes, positioning them close to the source of ATP and NADPH. Others are organized into metabolic complexes that channel intermediates directly from one enzyme to the next, preventing the loss of valuable metabolites. The Calvin cycle also demonstrates the remarkable efficiency that evolution can achieve given enough time. Despite the complexity of the pathway and the precision required for its operation, plants can fix carbon at rates of several hundred molecules of CO2 per second per molecule of Ru-bis-Co. The entire cycle from CO2 fixation to G3P formation can be completed in a few seconds under optimal conditions, allowing plants to rapidly convert atmospheric carbon into useful organic compounds when light and CO2 are abundant. The story of oxygen in Earth's atmosphere is intimately intertwined with the story of photosynthesis, a tale that unfolds over billions of years and fundamentally changed the nature of life on our planet. To understand this story fully, we need to journey back to Earth's early atmosphere, a time when the air contained virtually no free oxygen and life existed only in forms that would seem alien to us today. In Earth's earliest atmosphere, oxygen was not absent, but was locked up in compounds like water and carbon dioxide. Free molecular oxygen, O2, the gas we breathe, was essentially non-existent. The atmosphere was dominated by nitrogen, carbon dioxide, methane, and other gases that would be toxic to most modern life forms. This was actually a perfectly reasonable state of affairs from a chemical perspective, because free oxygen is extremely reactive and tends to combine with other elements rather than remaining free in the atmosphere. The earliest life forms were well adapted to this oxygen-free world. They were anaerobic organisms that obtained energy through fermentation and other chemical processes that don't require oxygen. Some of these early microbes actually found oxygen toxic because it could damage their cellular components through oxidation reactions. In many ways, the early Earth was their world, and they thrived in the oxygen-free conditions that prevailed for the first 2 billion years of our planet's history. The first photosynthetic bacteria that evolved didn't produce oxygen either. They used simpler forms of photosynthesis that relied on hydrogen sulfide or other electron donors instead of water. These early photosynthetic organisms were important pioneers, proving that sunlight could be harnessed as an energy source. But they didn't fundamentally change the chemistry of Earth's atmosphere. The real revolution began with the evolution of oxygenic photosynthesis in cyanobacteria, sometimes around 3.5 to 2.5 billion years ago. These remarkable microbes developed the ability to use water as an electron donor in photosynthesis, splitting H2O molecules to obtain the electrons needed for carbon fixation. The hydrogen from water was incorporated into organic compounds, while the oxygen was released as a waste product. At first, this oxygen didn't accumulate in the atmosphere. Earth's oceans and crustal rocks contained vast amounts of reduced iron that readily reacted with oxygen to form iron oxides. For hundreds of millions of years, the oxygen produced by cyanobacteria was immediately consumed by these geological sinks, preventing its accumulation in the atmosphere. During this time, banded iron formations were deposited in oceans around the world, alternating layers of iron-rich and silica-rich rock that record the gradual oxidation of oceanic iron. But eventually, around 2.4 billion years ago, the iron sinks became saturated, and oxygen began to accumulate in the atmosphere itself. This marked the beginning of the Great Oxidation Event, one of the most dramatic changes in Earth's history. Atmospheric oxygen levels rose from near zero to perhaps 1–2% of current levels over a period of a few 100 million years, rapid by geological standards, but still representing thousands of human lifetimes. This rise in atmospheric oxygen was catastrophic for many forms of life that had evolved in the oxygen-free world. Oxygen is a powerful oxidizing agent that can damage proteins, lipids, and other biological molecules through the formation of reactive oxygen species, free radicals that can wreak havoc in living cells. Many anaerobic organisms went extinct during this time, unable to cope with the toxic effects of the new oxygen-rich environment. Yet this apparent catastrophe set the stage for unprecedented biological innovation. Some organisms evolved sophisticated antioxidant systems to protect themselves from oxygen damage. Others went further and learned to harness oxygen for their own metabolic purposes, developing aerobic respiration, the process of using oxygen to extract energy from organic compounds. Aerobic respiration is far more efficient than anaerobic metabolism. While fermentation and other oxygen-free processes typically yield only 2 molecules of ATP per molecule of glucose, aerobic respiration can yield 32 molecules of ATP from the same starting material. This 16-fold increase in energy efficiency made possible the evolution of larger, more complex cells and eventually multicellular organisms. The rise in atmospheric oxygen also led to the formation of the ozone layer in the upper atmosphere. Ozone, O3, forms when ultraviolet radiation from the sun strikes oxygen molecules, causing some of them to combine into 3-atom molecules. This ozone layer acts as a shield, absorbing much of the harmful ultraviolet radiation that would otherwise reach Earth's surface. Before the ozone layer formed, UV radiation would have been lethal to most life forms on land, confining life to the protective depths of the oceans. With the ozone shield in place, life could begin to colonize terrestrial environments. The first land plants appeared around 450 million years ago, bringing photosynthesis to terrestrial ecosystems for the first time. These early land plants were simple, lacking the complex vascular systems and sophisticated structures of modern plants, but they established the foundation for all terrestrial ecosystems that would follow. The colonization of land by plants had profound effects on atmospheric oxygen levels. Terrestrial photosynthesis added to the oxygen production already occurring in marine environments, while the burial of organic matter in terrestrial sediments removed carbon from the active carbon cycle, indirectly leading to higher oxygen levels. By the Carboniferous period, around 300 million years ago, atmospheric oxygen levels had risen to as much as 35% of the atmosphere, significantly higher than today's 21%. These elevated oxygen levels had dramatic effects on life. The high oxygen concentration supported combustion much more readily than today's atmosphere, meaning that wildfires would have been more frequent and more intense. The abundant oxygen also supported gigantism in arthropods, insects, and other invertebrates that rely on passive diffusion for oxygen transport. Dragonflies with wingspans of over 2 feet and millipedes more than 6 feet long thrived in the oxygen-rich atmosphere of the Carboniferous. However, such high oxygen levels also created their own problems. The increased fire risk made it difficult for vegetation to establish in many areas, and the physiological challenges of dealing with highly oxidizing conditions may have stressed many organisms. Eventually, atmospheric oxygen levels stabilized at roughly current levels through a complex interplay of photosynthetic oxygen production, respiration and decay, and geological processes that bury or expose organic carbon. This stabilization represents a remarkable example of planetary-scale homeostasis, the ability of Earth systems to maintain relatively stable conditions despite changing inputs and outputs. The oxygen cycle is regulated by negative feedback mechanisms that tend to keep atmospheric oxygen levels within a relatively narrow range. If oxygen levels rise too high, increased fire frequency reduces plant biomass and oxygen production. If oxygen levels fall too low, reduced fire frequency allows more plant growth and increased oxygen production. As you settle deeper into peaceful rest, consider the extraordinary journey that every oxygen molecule you breathe has taken. Some of those molecules may have been produced by cyanobacteria, floating in ancient seas billions of years ago. Others may have been released just today by the tree outside your window, or by phytoplankton floating in distant oceans. Each molecule represents a moment when photosynthesis split a water molecule, liberating oxygen as a byproduct of the process that converts sunlight into the chemical energy of life. The oxygen in our atmosphere is not a permanent feature of our planet, but a dynamic component that is constantly being produced and consumed. Every breath you take removes oxygen from the atmosphere, while every moment of photosynthesis replaces it. The balance is so finely tuned that atmospheric oxygen levels remain remarkably stable from year to year. Despite the enormous fluxes of oxygen production and consumption that occur continuously around the globe, modern measurements show that atmospheric oxygen levels fluctuate slightly with the seasons, rising during the northern hemisphere summer, when photosynthesis is most active, and falling during winter, when photosynthesis slows, but respiration continues. These seasonal variations are tiny, less than 0.1% of total atmospheric oxygen, but they can be detected by sensitive instruments and serve as a reminder that our atmosphere is a living, breathing system maintained by the activities of countless photosynthetic organisms. The oxygen story also highlights the interconnectedness of life and Earth's physical systems. The evolution of oxygenic photosynthesis didn't just change biology, it changed geology, climate, and atmospheric chemistry. The formation of the ozone layer affected the penetration of ultraviolet radiation to Earth's surface. Changes in atmospheric composition affected the greenhouse properties of the atmosphere and thus global climate. The burial of organic matter in sedimentary rocks created fossil fuel deposits that would later be exploited by human civilization. Looking toward the future, the oxygen cycle continues to play a crucial role in maintaining Earth's habitability. Human activities are affecting this cycle in complex ways. Deforestation reduces photosynthetic oxygen production, while fossil fuel burning consumes oxygen and produces CO2. Climate change affects the distribution and activity of photosynthetic organisms, both on land and in the oceans. Understanding these interactions is crucial for predicting how our planet's life support systems will respond to continued human influence. The story of oxygen and photosynthesis reminds us that we live on a planet shaped by life itself. The air we breathe, the climate we experience, the very possibility of complex multicellular organisms like ourselves. All of these depend on the continuing activity of photosynthetic organisms that quietly and persistently convert sunlight into the chemical energy of life, while producing the oxygen that makes our existence possible. Beneath the familiar story of photosynthesis lies a deeper tale of partnership and cooperation that reveals the true interconnectedness of life on Earth. Plants don't photosynthesize in isolation. They exist within complex webs of relationships with other organisms, from the microscopic bacteria in their roots to the insects that pollinate their flowers, from the fungi that extend their reach into soil to the animals that disperse their seeds. These partnerships are so fundamental that photosynthesis, as we know, it simply couldn't exist without them. The most ancient and perhaps most crucial of these partnerships exists within every photosynthetic cell, a relationship so intimate that we often forget it's a partnership at all. Chloroplasts, the organelles where photosynthesis occurs, are not actually part of the original plant cell, but are descendants of ancient cyanobacteria that were engulfed by early eukaryotic cells billions of years ago. This endosymbiotic relationship, literally meaning living together inside, represents one of the most successful partnerships in evolutionary history. The evidence for this ancient partnership is written into the very structure and genetics of chloroplasts. Like their bacterial ancestors, chloroplasts have their own DNA, separate from the plant cell's nuclear DNA. They reproduce by dividing like bacteria, and their genetic code reveals unmistakable signs of their cyanobacterial heritage. The chloroplast genome contains genes for photosynthetic proteins, but many genes have been transferred to the plant cell nucleus over evolutionary time, creating a genetic interdependence that binds the partners together permanently. This partnership benefits both sides profoundly. The chloroplasts provide the plant cell with sugars and other organic compounds produced through photosynthesis, while the plant cell provides protection, structural support, and many of the proteins needed for photosynthesis. The plant cell can't survive without its chloroplasts, and the chloroplasts can't survive independently of their host cells. They have become so integrated that we think of them as plant organs rather than separate organisms. But their bacterial origins remind us that even the most basic processes of life often depend on cooperation between different forms of life. Above ground, plants engage in partnerships with countless other organisms that help them capture light more effectively or protect their photosynthetic apparatus from damage. Many plants host beneficial bacteria on their leaf surfaces, part of the phyllosphere, the ecosystem that exists on plant leaves. These bacterial partners can help protect plants from pathogens, provide nutrients, or even help process sunlight by breaking down complex organic compounds that might otherwise shade the leaf surface. Some plants have evolved partnerships with animals that help optimize their photosynthetic efficiency. Certain tropical plants have relationships with ants that help clear their leaves of debris and fungal growth, keeping their photosynthetic surfaces clean and functional. The ants receive shelter and sometimes food from the plants, while the plants benefit from the ants' housekeeping services that maintain optimal conditions for photosynthesis. Below ground, an even more extensive network of partnerships supports the photosynthetic activities happening above the surface. Plant roots form intimate associations with soil microorganisms that dramatically expand their ability to obtain the nutrients needed for photosynthesis. The most widespread of these partnerships involves mycorrhizal fungi, literally fungus roots, that form connections with the roots of most land plants. Mycorrhizal fungi extend the effective reach of plant root systems by hundreds or even thousands of times. Their microscopic filaments, called hyphae, spread through soil much more extensively than plant roots can, exploring tiny spaces between soil particles and extracting nutrients that would otherwise be unavailable to plants. In return for these nutrients, particularly phosphorus and nitrogen, that are essential for photosynthetic proteins, The fungi receive sugars produced by plant photosynthesis. This underground partnership is so extensive and so important that some scientists describe it as a wood wide web, a network of fungal connections that links plants across entire forests. Through these fungal networks, plants can share resources, with trees sending surplus sugars to smaller plants growing in their shade, and nutrient-rich areas of soil supporting plants in nutrient-poor areas through the fungal network. The fungi can even carry chemical signals between plants, warning of insect attacks or environmental stresses. Some plants have evolved even more specialized root partnerships with bacteria that can fix nitrogen from the atmosphere. Legumes like beans, peas, and clover form nodules on their roots that house nitrogen-fixing bacteria. These bacteria have the remarkable ability to convert atmospheric nitrogen gas, which makes up 78% of our atmosphere, but is chemically inert and unavailable to most life forms, into ammonia that can be used to make amino acids and proteins. This nitrogen fixation process requires enormous amounts of energy, and the bacteria depend entirely on sugars produced by plant photosynthesis to power their nitrogen-fixing machinery. In return, they provide the plant with a private source of nitrogen fertilizer, giving legumes a significant advantage in nitrogen-poor soils. This partnership is so efficient that legumes can actually improve soil fertility, which is why they're often used in crop rotation systems to restore nutrients to agricultural fields. The energy cost of supporting nitrogen-fixing bacteria is substantial. It can consume 20% or more of the photosynthetic products produced by the plant. But this investment pays off handsomely in environments where nitrogen is limiting plant growth. The partnership allows legumes to thrive in soils where other plants struggle, and the excess nitrogen they produce benefits other plants in the ecosystem as well. As you rest in the gathering quiet, imagine the invisible networks of cooperation that surround every plant. Fungal threads are extending through soil, connecting root systems and sharing nutrients over distances measured in meters or even kilometers. Bacteria in root nodules are splitting nitrogen molecules and combining them into organic compounds. Beneficial microbes on leaf surfaces are protecting photosynthetic tissues from harmful pathogens and environmental stresses. These partnerships extend far beyond individual plants to shape entire ecosystems. In many forests, the trees, understory plants, and even non-photosynthetic organisms like mushrooms are connected through vast networks of mycorrhizal fungi. These networks can persist for decades or centuries, maintaining connections between plants across generations and helping ecosystems recover from disturbances like fires or storms. The partnerships that support photosynthesis also extend into the realm of reproduction and genetic exchange. Most flowering plants depend on partnerships with animals for pollination. The transfer of pollen from male to female flower parts that allows sexual reproduction. These partnerships are often exquisitely co-evolved, with flowers providing nectar, pollen, or other rewards to animal partners in exchange for pollination services. The energy for producing these floral rewards comes directly from photosynthesis, representing another investment of photosynthetic products in partnership relationships. Some plants invest enormous amounts of energy in large, showy flowers that attract pollinators from great distances. Others produce sweet nectar that provides high-energy fuel for their pollinator partners. The diversity of flower shapes, colors, and scents reflects the diversity of these pollination partnerships, each one fine-tuned by evolution to attract specific animal partners. Even seed dispersal often involves partnerships between plants and animals, with plants producing nutritious fruits that animals eat in exchange for carrying seeds to new locations. The energy to produce these fruits comes from photosynthesis, representing yet another way that the products of light capture are invested in relationships that benefit plant survival and reproduction. These partnership networks reveal that photosynthesis is not simply a plant process, but an ecosystem process that involves countless species working together. The oxygen you breathe was produced not just by plants acting alone, but by plants working in partnership with their bacterial, fungal, and animal partners. The carbon captured from the atmosphere flows through complex networks of exchange that link photosynthetic organisms with the entire web of life. Understanding these partnerships also helps us appreciate how human activities affect photosynthesis and ecosystem function. When we disrupt soil communities through intensive agriculture or pollution, we're not just affecting soil, we're disrupting the fungal and bacterial partnerships that support plant photosynthesis. When we reduce pollinator populations through habitat loss or pesticide use, we're affecting the reproductive success of the plants that capture carbon from our atmosphere. Conservation efforts increasingly recognize the importance of these partnership networks. Protecting photosynthetic ecosystems means protecting not just the plants themselves, but the entire communities of organisms that support them. Restoration efforts that focus only on replanting trees or grasses often fail because they don't restore the soil communities and partnership networks that those plants depend on. The story of photosynthetic partnerships reminds us that cooperation, not just competition, is a fundamental force in evolution and ecology. The most successful organisms are often those that have evolved the most beneficial partnerships, and the most productive ecosystems are those where these partnership networks are most fully developed. In the quiet, patient work of photosynthesis, we see not just individual organisms capturing light, but entire communities working together to transform solar energy into the chemical energy that powers the web of life. As we settle into the deepest part of our journey through the world of photosynthesis, it's time to contemplate how this ancient process might unfold in the future, both on Earth and perhaps beyond. The story of photosynthesis is far from over. It continues to evolve, adapt, and inspire new possibilities that stretch our imagination, while remaining grounded in the patient, persistent chemistry that has sustained life on our planet for billions of years. On Earth, photosynthesis faces new challenges and opportunities as our climate continues to change. Rising atmospheric carbon dioxide levels are already affecting photosynthetic organisms in complex ways. For many plants, higher CO2 concentrations provide more raw material for the Calvin cycle, potentially increasing photosynthetic rates and plant growth. This CO2 fertilization effect has been observed in many ecosystems, with some plants growing larger and faster than they have in thousands of years. But the story is more complex than simple fertilization. As atmospheric CO2 continues to rise, many plants are becoming limited by other factors, particularly the availability of nitrogen and phosphorus needed to build new photosynthetic machinery. The partnerships with mycorrhizal fungi and nitrogen-fixing bacteria become even more critical as plants try to take advantage of increased CO2 levels. Some plants are adjusting by changing the ratio of different proteins in their leaves, reducing the amount of Rhu-bis-Co, and increasing enzymes involved in other aspects of metabolism. Temperature changes are also affecting photosynthesis in ways that vary dramatically across different ecosystems and plant types. In some cold-limited environments, warming temperatures are extending growing seasons and allowing photosynthesis to continue longer into the fall and begin earlier in the spring. Arctic tundra regions are seeing dramatic increases in plant growth as temperatures rise and permafrost thaws, potentially turning these regions from carbon sources to carbon sinks. In other regions, rising temperatures are beginning to stress photosynthetic systems beyond their optimal operating ranges. When temperatures get too high, the enzymes involved in photosynthesis begin to lose their precise three-dimensional structures, reducing their efficiency. Plants must divert more energy into cooling mechanisms and stress responses, reducing the net gain from photosynthesis. Some plants are responding by adjusting their daily rhythms of photosynthesis, becoming more active during cooler morning and evening hours. Ocean photosynthesis is facing particularly complex challenges as seawater becomes warmer and more acidic due to climate change. Many marine photosynthetic organisms are highly sensitive to temperature changes, and warming oceans are already causing shifts in phytoplankton communities toward species better adapted to higher temperatures. Ocean acidification, caused by the absorption of excess atmospheric CO2 into seawater, is affecting the ability of some marine organisms to build calcium carbonate shells and structures, potentially disrupting marine photosynthetic ecosystems. Yet photosynthetic organisms have shown remarkable resilience and adaptability throughout Earth's history. They have survived ice ages, volcanic eruptions, asteroid impacts, and dramatic changes in atmospheric composition. Many researchers believe that photosynthetic life will continue to adapt to changing conditions, perhaps in ways that surprise us. Some plants are already evolving improved heat tolerance, more efficient water use, and better ability to photosynthesize under variable light conditions. Human technology is also beginning to learn from and work with photosynthetic systems in new ways. Biomimetic research, the science of learning from nature to improve human technology, is producing new solar energy systems inspired by the molecular machinery of photosynthesis. These artificial photosynthetic systems aim to capture and convert solar energy with the efficiency and elegance of natural photosynthesis, potentially providing clean energy solutions for human civilization. Some of the most promising developments involve hybrid systems that combine biological and artificial components. Researchers are developing systems that use real photosynthetic proteins embedded in artificial membranes to capture light energy, potentially achieving the efficiency of biological systems while providing the durability and controllability of engineered systems. Other approaches use photosynthetic microorganisms as living solar cells, harvesting the electrons they produce during photosynthesis to generate electricity. Agricultural research is working to develop crops with improved photosynthetic efficiency, potentially allowing farmers to produce more food with less land, water, and fertilizer. Scientists are exploring ways to introduce C4 photosynthesis into rice and other C3 crops, potentially increasing their efficiency in hot, dry conditions. Other research focuses on improving the efficiency of Rubisco, or developing alternative carbon fixation pathways that avoid the wasteful process of photorespiration. Genetic engineering and synthetic biology are opening new possibilities for modifying and enhancing photosynthetic systems. Researchers are working to develop plants that can photosynthesize more efficiently in low light conditions, potentially allowing agriculture to expand into marginal areas, or to use artificial lighting more effectively. Others are engineering plants to produce useful compounds, pharmaceuticals, biofuels, or industrial chemicals directly through modified photosynthetic pathways. As you rest deeply in the quiet darkness, consider that the future of photosynthesis might extend far beyond our own planet. As humans begin to explore and potentially inhabit other worlds, we may bring photosynthetic life with us as essential partners in creating habitable environments. Plants and photosynthetic microorganisms could be crucial components of life support systems on space stations, lunar bases, or Mars colonies, providing oxygen, food, and psychological benefits to human explorers. The challenge of photosynthesis in space environments is pushing researchers to understand these systems in entirely new ways. How would plants photosynthesize under the different light conditions on Mars, where sunlight is dimmer and filtered through a different atmosphere? Could we develop photosynthetic organisms adapted to the low-gravity environments of the Moon or asteroids? Might we discover forms of photosynthesis on other worlds that use different wavelengths of light or different chemical pathways? Some researchers speculate about the possibility of terraforming, transforming the atmospheres of other planets to make them more Earth-like. Photosynthetic organisms could potentially play a role in such massive engineering projects, gradually converting atmospheric CO2 to oxygen over geological timescales, just as they did on early Earth. The timescales involved would be enormous, thousands or millions of years. But the processes would be fundamentally the same as those occurring quietly in every leaf and algae cell today. Even more speculatively, the discovery of life on other worlds might reveal entirely different approaches to photosynthesis. Life on planets orbiting different types of stars might have evolved photosynthetic systems that capture different wavelengths of light. The basic chemistry might be similar, capturing photons to drive chemical reactions that store energy. But the specific molecular mechanisms could be unlike anything we see on Earth. The search for life on other worlds often focuses on finding signs of photosynthesis, particularly the presence of oxygen in planetary atmospheres that could only be maintained by biological activity. The James Webb Space Telescope and other advanced instruments are beginning to analyze the atmospheres of distant exoplanets, looking for the chemical signatures that might indicate photosynthetic life. Finding such signatures would transform our understanding of life in the universe and potentially reveal new possibilities for photosynthetic chemistry. But perhaps the most profound future developments in photosynthesis will come from simply continuing to understand and appreciate the systems we already have. Every year, researchers discover new aspects of photosynthetic function, new species with unique adaptations, new relationships between photosynthetic organisms and their environments. The more we learn about photosynthesis, the more we realize how much we still don't know about these fundamental processes of life. The future of photosynthesis is ultimately the future of life on Earth and perhaps beyond. As long as stars shine and planets receive their light, there will likely be opportunities for photosynthetic organisms to capture that energy and transform it into the chemistry of life. Whether through natural evolution, human enhancement, or expansion to other worlds, photosynthesis will continue its patient, persistent work of turning light into life. In our own time, we have the privilege of understanding photosynthesis more deeply than any generation before us, while also recognizing how much mystery remains. Every breath we take connects us to the ancient partnership between light and life. Every meal we eat traces back to photosynthetic chemistry. Every moment of our existence depends on the continuing work of photosynthetic organisms around the globe. As our journey through the world of photosynthesis draws toward its peaceful close, we find ourselves returning to where we began, to the quiet miracle that surrounds us every moment. the gentle revolution that never stops, the patient transformation of light into life that has been happening for billions of years and continues still in the darkness outside your window. Photosynthesis teaches us about time in a way that few other natural processes can. It operates simultaneously on timescales that span from the quantum mechanical electrons, moving through transport chains in picoseconds, to the geological, atmospheric oxygen levels, changing over hundreds of millions of years. In between these extremes, photosynthesis marks the rhythms of daily life. The opening of stomata with the morning sun, the seasonal cycles of growth and dormancy, the patient accumulation of biomass that builds forests and fills grasslands. There's something profoundly comforting about this temporal continuity. While human affairs often seem urgent and chaotic, photosynthesis proceeds with an unhurried persistence that has sustained life through every crisis our planet has faced. Ice ages come and go, continents drift across the globe, species arise and disappear. But the fundamental chemistry of light and life continues unchanged. Chlorophyll molecules still capture photons with the same efficiency they achieved billions of years ago. The Calvin cycle still fixes carbon with the same elegant choreography. Water molecules still split to release oxygen with the same gentle persistence. This continuity doesn't mean photosynthesis is static or unchanging. Evolution continues to refine and adapt photosynthetic systems, finding new solutions to environmental challenges, developing new partnerships with other organisms, exploring new ways to capture and utilize light energy. But the changes happen gradually, each modification building on the solid foundation of chemistry and physics that makes photosynthesis possible. In our fast-paced world, there's wisdom in contemplating the patience of photosynthesis. Plants don't rush their growth or hurry their development. They respond to the availability of light, water, and nutrients, but they don't try to force conditions to change faster than they naturally would. A tree doesn't worry about next year's rainfall or next decade's temperature changes. It simply continues the daily work of photosynthesis, building its structure carbon atom by carbon atom, day by day, season by season. This patient persistence has allowed photosynthetic organisms to colonize virtually every environment on Earth where light penetrates. From the frigid polar regions where plants photosynthesize under the midnight sun, to the scorching deserts where cacti capture carbon in the cool of night. From the crushing depths of ocean trenches where chemosynthetic bacteria process chemicals in eternal darkness, to the heights of mountains where alpine plants photosynthesize in thin air and intense ultraviolet light, life has found ways to capture and utilize available energy. The diversity of photosynthetic strategies reflects the fundamental creativity of evolution when given enough time to explore possibilities. C3, C4, and CAM photosynthesis represent different solutions to the challenge of capturing carbon while conserving water. Shade plants and sun plants have evolved different approaches to capturing light efficiently under high and low light conditions. Arctic plants and tropical plants have adapted their photosynthetic machinery to function optimally at very different temperatures. Yet beneath this diversity lies the fundamental unity of photosynthetic chemistry. Whether in the smallest bacterium or the largest tree, the basic process remains the same. Light energy excites electrons. Those electrons power chemical reactions that split water and fix carbon. And the products of those reactions provide the energy and materials needed for growth, reproduction, and all the other processes of life. This unity reminds us of our deep connection to all photosynthetic life. The carbon in our bodies was once floating in the atmosphere as CO2, until some photosynthetic organism captured it and incorporated it into organic compounds. The oxygen in our blood was produced when some plant, somewhere, split a water molecule using the energy of sunlight. Every meal we eat ultimately traces back to photosynthesis, whether directly from plant foods or indirectly through animals that fed on plants. As you sink deeper into rest, let your breath become slow and regular, and with each exhale, remember that you're returning CO2 to the atmosphere, where it will eventually be captured again by photosynthesis. With each inhale, you're taking in oxygen that was released by photosynthesis as a gift from the plant world to all aerobic life. This exchange has been happening with every breath you've ever taken, connecting you intimately to the global cycles of carbon and oxygen that photosynthesis maintains. The beauty of photosynthesis extends beyond its chemistry to its role in creating and maintaining the conditions that make Earth habitable. Without photosynthesis, our planet would have no free oxygen, no ozone layer, no complex ecosystems, no fossil fuels, no soil organic matter, no forests or grasslands or coral reefs. The entire biosphere, as we know it, is ultimately a product of photosynthesis, built up over billions of years through the patient accumulation of photosynthetic products. This perspective can help us appreciate both the robustness and the fragility of the systems that support life. Photosynthesis is robust because it has survived and thrived through enormous environmental changes over geological time. It's fragile because it depends on precise molecular machinery operating under specific environmental conditions. Small changes in temperature, light, or chemistry can have large effects on photosynthetic efficiency, and disruptions to photosynthetic ecosystems can cascade through entire food webs. Understanding photosynthesis also provides perspective on human sustainability and our relationship with the natural world. Human civilization depends entirely on photosynthetic products, not just for food, but for fiber, fuel, construction materials, pharmaceuticals, and countless other necessities. Our fossil fuels are ancient photosynthetic products, buried and concentrated over millions of years. Our agricultural systems are essentially managed photosynthetic ecosystems designed to maximize the production of useful organic compounds. Yet we often take photosynthesis for granted, treating it as a free service provided by nature, rather than recognizing it as the fundamental process that makes our existence possible. Climate change, deforestation, soil degradation, and pollution all threaten photosynthetic systems in ways that could ultimately threaten the conditions that support human civilization. Learning to work with photosynthetic systems rather than against them may be one of the most important challenges facing our species. The story of photosynthesis offers hope as well as warning. If life could transform Earth's atmosphere from oxygen-free to oxygen-rich over the course of billions of years, then perhaps human intelligence and technology, working with natural processes rather than against them, could help address the environmental challenges we face today. Biomimetic technologies inspired by photosynthesis might provide clean energy solutions. Agricultural systems that work with natural photosynthetic processes and partnerships could feed growing populations while protecting ecosystems. Understanding photosynthetic ecosystems could help us design cities and landscapes that support both human needs and natural processes. The contemplation of photosynthesis also offers a different way of thinking about success and productivity. In human society, we often value speed, growth, and dramatic achievement. Photosynthesis suggests alternative values: patience, persistence, and the quiet accumulation of small gains over long periods. A forest doesn't achieve its majesty overnight, but through decades or centuries of steady growth. A soil doesn't become rich and fertile quickly, but through the gradual accumulation and decomposition of organic matter over many years. There's a meditative quality to watching photosynthesis at work, the slow movement of leaves tracking the sun across the sky, the gradual unfurling of new growth in spring, the patient process of fruits ripening as they accumulate the products of photosynthesis. These natural rhythms operate on timescales that encourage contemplation and reflection, inviting us to slow down and appreciate processes that can't be hurried or forced. As the night deepens around you and your breathing slows into the rhythm of peaceful sleep, imagine yourself connected to the vast, quiet network of photosynthetic activity that spans our planet. In the darkness, some plants are using stored energy to grow and repair their photosynthetic machinery, preparing for tomorrow's return of sunlight. In other parts of the world where the sun is still shining, billions of chloroplasts are quietly capturing photons and converting them into the chemical energy that sustains the web of life. This network has no central control, no conscious direction, no master plan. Yet it maintains the atmospheric composition that makes life possible, regulates global climate through its effects on carbon and water cycles, and provides the foundation for all terrestrial and most marine ecosystems. It's a testament to the power of simple, repeated actions accumulating over vast scales of time and space. The gentle revolution of photosynthesis continues all around us and within us, as constant and reliable as sunrise, as fundamental as the laws of physics, as beautiful as the play of light through leaves on a summer afternoon. In understanding this process, we touch something essential about the nature of life itself: its patience, its persistence, its ability to transform simple materials into endless complexity and beauty. As you drift towards sleep, carry with you the quiet wonder of photosynthesis, the knowledge that every breath connects you to ancient bacterial partnerships, that every bite of food traces back to sunlight captured by plants, that every moment of your existence is sustained by the ongoing transformation of light into life. In this understanding, sleep becomes not just rest, but a peaceful participation in the great cycles that connect all living things, a gentle settling into the rhythm of a world powered by light, sustained by patience, and united by the chemistry of life itself. Rest well in the knowledge that the gentle revolution continues, as it has for billions of years, as it will for billions more, quietly and persistently transforming our world one photon at a time, one molecule at a time, one breath at a time. The ancient partnership between light and life cradles you in its patient embrace. And in the morning, when sunlight returns to wake the world, photosynthesis will resume its quiet work of turning starlight into life, just as it always has, just as it always will.